Okay, so the first thing I want to mention is I sent home a little email with your marks and whether or not you had any missing assignments or quizzes yesterday. Something to keep in mind with regards to that. That is still an early mark. It is the mark I normally would have released in the middle of last week. So please keep in mind that there are still a lot of assessments left to go onto your mark. A lot. So the weighting of each specific assessment, especially the summative assignments at the end of the units, is like extremely high. So if you did very poorly on one particular assignment, it may have like, at least as of now, tanked your mark. Okay, so keep that in mind. It might look really bad, but that is one assignment, right? You could do really well on the next assignment, your mark could go up 25%. Okay, so when I post such an early mark, it's very tentative. There's so much more to add to it, and so every item is worth so much. One minor thing could potentially really affect your mark. Okay. Also, something to keep in mind that when I give you your final mark, I look at the most recent and consistent performance in the course. Okay, so if you got 95 on every quiz, but you had a quiz where you got 65, I usually just ignore that quiz. I just like take it off your mark because that's not your consistent performance in the course. Okay, everybody has a bad day, everybody has a bad quiz, everybody has an assignment where they're like, whoops, I messed this up, or I didn't do this thing quite right and I didn't understand it properly, or whatever. Okay, that's not really how we're supposed to be compiling your marks at the end of any semester. We're supposed to be looking at what your consistent performance is with a emphasis on what the most recent performance is. So just something to keep in mind. Like, don't think that one item, if it didn't go well right now, and I don't do that, by the way, for this mark. This mark is just a mathematical computation of your current course grade with weightings taken into account. But it's just a mathematical grade. So I haven't looked at it and been like, this is out of character for this person and like eliminated stuff. Not at this point in the course, it's too early. But just keep in mind that I do that at the end. Okay, so if you have a really bad quiz or something, it's not going to tank your mark at the end of the semester. There are a number, don't forget to check the comments on the assignments when I return them to you. Sometimes I ask people to resubmit things, okay? If I feel like you're way off on the assignment, you've handed something in and you like did not understand some key components of the assignment, I might ask you to resubmit it. You don't have to. Okay, that stuff's always voluntary, but you have the opportunity to resubmit it because I'd like to see whether or not, you're capable of doing those particular components of the assignment, and maybe you like missed the boat on it, like misunderstood what you were supposed to do. And like, again, that's not the purpose of it. I'm not trying to get you on the assignments. I'm just trying to assess your ability to complete the components of each assignment and like demonstrate your knowledge of the course material. Not necessarily like to just like to get you for like, oh, you didn't get this, I gotcha. Like that's not the purpose of the assessment. Okay, I want everybody in here to be successful. So if you're in that boat, I may have asked you to resubmit. You don't have to. Okay, it's always optional, but maybe you choose that. And if you do, don't be afraid to email me and be like, I'm not 100% sure on what this comment means, or I got to improve this. I'm not sure what that means. Just let me know. Okay, like I'm happy to talk to you about that. I want you to do well. So. I just wanted to point that out because I did get some emails from people that are like, how come my mark is this or whatever? It's probably because of one item. So don't take that super to heart yet. There's a whole bunch of other opportunities. We got all the summative stuff from this unit, etc. I'll email you back anyway, but just to be clear on that, I don't want people to freak out this early in the course. I mean, right, it's not that early in the course anymore, but in terms of where the marks are, it's early. So. Okay, that being said, let's talk about, did anybody have any questions from Friday's content? Stems and roots. You finished the roots component. Maybe you were just like, oh, I just didn't get this thing about roots. Anybody in that boat? No. Roots are pretty straightforward. Okay, cool. That's fine. Let's look at what we're doing today then. It always tries to scan my face when I log in, but it doesn't work with my mask. Come on, just skip it. Sorry. Try and get this set up before we get started. It's kind of a pain. In order to get my iPad to work, I have to set up a mobile hotspot on my laptop and then connect to it with my iPad. Otherwise, I can't mirror my iPad on here, which is kind of a pain. It's going to work now. Yay. Yeah? There we go. All right, some of you may have noticed I brought some flowers in. The grocery store was really not great this time looking for flowers. I wonder if it's time of year. Maybe not a lot of people are buying flowers this time of year. This is what I could get. It's not a great selection. I would have bought 2 of the first one there. Those are like daylilies, but because they are perfect flowers. They have male and female parts. I'll show you what they look like in a second. But I got these other standard lilies over here, which are usually great. And I tried really hard over the weekend to get them to bloom. You're supposed to like blow on them with a hair dryer and put them in the sun. It didn't work. I just could not get them to bloom. So we can still cut them open and look at the bloom inside it. It's in there, but it's just not as nice. But anyway, we got to work with what we got here. So I had to go to a few grocery stores actually to find them. So I don't know. I guess flowers are not really big right now. Maybe it's just the grocery stores in my area. I'm not sure. But oh, I forgot to change the link for that. I got to fix that. So normally I do take a course survey around this time, but this is actually the course survey from last quad. I'm gonna fix that. I'll actually post it for tomorrow. I do it at the end of this block usually. Forgot to remove that. So two things we're going to cover today. You've done the anatomy of a plant, basically the roots, the stem, the leaf. That's the main parts of most plants. The second block today, we're going to do flowers. So we're going to look at the sexual reproductive organs of the plant, and we'll talk a little bit about fruit. But in between there, now that you know the basic anatomy of the different parts, we're going to talk about transport. So how do plants get stuff from one part of the plant to the other part of the plant? And there are 2 main things that the plant transports. One of them is water and water-soluble minerals and things like that. And those are transported in the xylem. And then you've got the sugar transport, which is occurring in the phloem. Did I get that correct? Yeah, that's right. Yeah, sugar transport in the phloem. So those are completely different and separate systems, although they do interact with one another a little bit. But one of them is almost completely a passive system. That's the water transportation mechanism in the xylem. So in the little lesson, which is down here, I go through how that works. But they're basically straws. They're empty tubes inside the plant. And it's mostly passive forces that are acting on the water to draw it up inside. The evapotranspiration, the evaporation of water off of the leaf, actually plays a large role in pulling water up through the xylem, which is kind of cool. There is a great video here on Veritasium, which talks about the physics behind how those mechanisms actually work, because they're really cool. And when you think about it, a tree, which could be like potentially 100 meters tall, okay, a really large structure, has to do some really cool physics in order to actually pull water up a tube of that size. If it was simply a pipe, like a water pipe, and you wanted to pull water up of a water pipe, it can't be done just purely by pulling it from the top. It's not like the physics of it are impossible. If you create a perfect vacuum at the top of the pipe, that is, you're basically pulling on the air at the top as hard as you can, that's not enough force to actually pull water up the pipe. And they go through all the physics of it here in the video. So there are a bunch of other mechanisms that the plant has to work with in order to actually get the physics of it to be possible to pump water up that high, which is really cool. There are 3 main mechanisms. I go through them in the note, but you need to know what those 3 mechanisms are, just so you know. Okay, so that's what we're getting at for the water component. All right, there's 3 mechanisms that are important. It talks in here just a touch about sugar transport, but that's not the real interesting thing with regarding the physics. Okay, the water transport is the cool physics game that trees play. Great video, by the way, and it does a great job of explaining that. I go through more of the physiology in the note part, like what do the cells actually look like, and like how do they grow and function and things like that regarding xylem and phloem. But and then the sugar component is completely different because it's bidirectional. Sometimes with water, you just want to get the water from the roots to wherever you're doing photosynthesis, because that's what you need the water for, right? To actually do photosynthesis. So the water is always going up. There's no downward pumping of water or downward movement of water. But sugar needs to go wherever the plant needs to send sugar. You want to store it in the roots, you got to pump it down. You want to bring sugar up into the leaf area so you can build new leaf, because you need the energy to build new cells or whatever, or to the apical meristem, which is the top of the plant where the most growth is occurring. You got to pump sugar up there. So sugar is like you basically want to send the sugar wherever you need it in the plant. So it's not a passive mechanism. You have to pump it. You have to physically take it and put it in the places where you want to put it. And so there are some mechanisms at play for transporting sugar from where it's being produced or where it's being stored to where you want it to go. Okay, so it works quite different from the water transportation mechanisms. And then you need to have a general sense of that process as well, and what types of cells are involved. Okay, there, the xylem cells have a specific name. The phloem cells have a specific name. The phloem cells have support cells that are next to them. You need to know what those are, etc. Okay, so the basics of how this process works for each. So that's what I'm going through in the note. It's all about transportation. And then there are some questions from the textbook. It's 571-4, and a little exit quiz. Again, basic stuff about transportation of plants. So that's block A. In block B, we're going to talk about sexual reproduction. So how does a plant primarily make baby plants? Okay, most plants are reproducing primarily through sexual reproduction. We're going to focus on flowering plants because they're the coolest, and well, at least I think they're the coolest. There are other strategies. I can't remember. I think we touch on gymnosperms a little bit, which are coned plants. It's similar. So the main difference between what gymnosperms are doing and flowering plants is that gymnosperms usually just cast their pollen into the air. If you've ever parked a car, do you guys drive yet? What are you guys, 16? So maybe you're driving a car. If you ever park under like a pine tree or something like that in the spring, don't recommend it, your car gets covered in pollen. You may have noticed this. And that's because that's the gymnosperm strategy for pollination, which is just throw a huge amount of pollen into the air and just hope that it lands on a neighboring tree. So the pollen has like these little air sacs to help it kind of fly through the air, and it just casts off tons and tons of pollen, a huge, huge quantity of pollen, which is very energy intensive for the plant. It has to produce a ton in order to actually hit its target, because it's just basically throwing it up in the air and hoping it lands in the right spot. So that's one strategy. That's the gymnosperm strategy. The flowering plant has a really cool strategy, which is just get another organism to take your pollen from your male part and just drop it off at a female flower. Cool. So those are pollinators. Most of the plants you're familiar with are probably angiosperms. They're flowering plants. And they use pollinators like bees to transport their pollen from plant to plant, which is way, way less energy intensive. You do have to lure in the insects. So there's an energy involved there. You got to produce some nectar or something for the insects to eat so that you can lure them in. But the payoff is that you have to produce way, way less pollen, which is the really labor-intensive part. That's the part you need to do meiosis for. And that meiotic process is very energy-intensive. So the less meiosis you have to do, the more energy you save. And saving energy just means better survival for you. So anyway, they use pollinators. So that's the main difference there. I'm not really going to talk about moss, pteridophyte, and pteridophytes and the other ones, ferns. Great. Another word's going to escape me here. Bryophytes are the mosses, and pteridophytes are ferns. There we go. Those two. They have a cool system, similar, but obviously they don't have pollinators involved. And they're pretty much just dropping their gametes into water and then just hoping that they're going to meet up with gametes from the other sex in the water. That's basically what happens for them. It's a simplified process. We're going to focus in on flowers, okay? Because again, I think they're the coolest. But it's also the curriculum focuses on flowers, and that's fine too. So we're going to dissect a flower together. We'll look at the parts, find the male, find the female, talk about the anatomy of each. Hopefully at home people have a flower. If not, it's not a huge deal. I'm gonna do one live here too, so you'll be able to see everything. But you're gonna do a little bit of reading on sexual reproduction and the actual cycle of it. Some of you guys are familiar with it already from your species spotlight assignments. If you picked a plant, a number of you I've seen have already gone through the life cycle for a flowering plant. Cool. You're just gonna see it again then. And the second component of this is I have an awesome documentary called The Power of Flowers. It's just entertaining, and it goes through the evolution of the flower. Why, over time, did plants transition from being primarily gymnosperms? So if you look 65 million years ago, dinosaur times, and you look around, it's all gymnosperms. There's no flowering plants in existence then. So at that point, it's like 95-ish% of all plants were gymnosperms. And now it's completely switched, where 90-ish% of plants are now angiosperms, and gymnosperms are actually quite rare. I mean, if you look at the overall plants out there, species, they're quite rare. So what happened? Why was there such a huge transition to flowering plants? And the video talks about that and talks about the history of seeds and all kinds of cool stuff. So it's a great documentary. There's is there a sheet that goes along with it? There might not be for this one. Let me look at the notes package here. I'm actually not sure if there's a sheet for this. Honestly, it doesn't really need a sheet. It's a fascinating documentary. There's transport, flowers. No, I guess it doesn't. Anyway, there's no sheet, but it's fascinating. So you won't really need the sheet to help focus, I don't think. There's a couple questions from the text and an exit quiz. Okay, the actual dissection itself takes 10 minutes. I mean, flowers aren't the most complicated structure you're ever going to see, but we are going to go through the parts together. There is a sheet that you fill in as you go through where you just write down the function of each part. All right? And that's it for today. So today we're covering transport and sexual reproduction. Tomorrow we're going to look at the other way that plants can reproduce, and that's asexually. So I'm sure you guys have heard of this before, that you can like take a cutting from a plant. You can grow a new plant from the cutting. This is probably not news to everybody. But that's a form of asexual reproduction. It's cloning. And there's a number of different ways that plants can asexually reproduce. We'll talk about them tomorrow. And then we'll work on the plants assignment, which I'm hoping I went over already. I think I did. Yes? Okay, cool. And then I'm going to give you the second half to work on the plants presentation. And then we're done plants, guys. And we're going to move in on Wednesday to our very last unit, which is evolution, kind of taking it all together, putting all the stuff from the different units together, genetics, and just putting it together and structure and function and putting it all together into evolution. A little introduction on Wednesday. And then we have this weird fractured schedule to the end. Sorry about that. Any questions before you get started? Okay, so there's a video that goes along with transport for block 1 here, or A block, or whatever we're calling it, the first block. And then I'm going to come back and we'll do the dissection together in block 2. Let me know if you need anything. Okay, before we get going with the flower stuff for this afternoon, or whatever, second block, does anybody have any questions from any of the transport stuff from this morning? Lots of new vocabulary in there, sieve tube elements and such. No? Fairly straightforward, I guess. Okay. Cool. No worries. If something comes up later, let me know. So, all right, let's just take a look at these together. Let me get set up here quick. I've got two, this was mentioned by the people in the class already, but we have two different flowers which we're looking at. These, which have unfortunately not opened yet, but we're still going to cut open one of the buds to take a look at the flower inside. It's still fully formed. And these. And they are both monocots, and as mentioned, the major clue that you get for that is if you look at the leaves. It's a little bit hard to see on the camera here, but they have parallel leaf veins, and parallel leaf vein is monocot. So transfer my camera over here. We figured out how to get that light working, didn't we?
Yeah, you just put it on video mode.
Video. Right, video mode. That's what it was.
Do the flash thing.
I do, yes. That goes on. Oh, great. There's no 2x zoom with this, unfortunately. It only works with the photo mode, but sorry, I think I got this little magnifying lens. Oh. Where? Flip this. Oops. Like this. Is this good enough? I actually, this is probably fine. Key is I need to be able to pull this little magnifying lens in front. That way, get up close and personal with some of this stuff. I'm just going to quickly clean this lens. It's dirty. Really dirty. There we go. So we'll be able to see inside some of the plant structures that are a little bit on the smaller side. Now, is this like upside down? No, this is okay. All right. You guys see that okay? Yeah? All right. Okay. I'm going to start with the daylily that I've got here. Use this white backdrop to make it a little clearer. there's our flower. This is an angiosperm, and as mentioned, this is specifically a monocot. Dicots have similar flowers, obviously. I mean, they're flower-like. If you're looking at just the flower, you can often tell if they're a monocot or a dicot. Monocots have 3 and multiples of 3 for their petals, I believe. Is that right? Double check this. Yeah. So multiples of 3 for petals. And if you look at the stamens, which are the male parts inside, write those out in here. They're all the ones that have an anther on top, which has a pollen sac in it. These right here. So we've got a filament. The filament is like the long tube that's attached to it, and then the anther is on top. It has the pollen sac. You can even see when I'm touching it, there's like little pieces of pollen falling off of it. So you also notice that they're pretty loose. They're like really loosey-goosey on the top there. And that's intentional. They're supposed to shake and jiggle and stuff like that when pollinators walk over top of them, so that it brushes the pollen onto the pollinator's body. You can even see there's some pollen on the inside of this flower that's fallen off of the anthers. So if you look at them, they should be in multiples of 3 because this is a monocot, unless one of them's fallen off. So 1, 2, 3, 4, 5, 6. So this one has 6. Yours should have a multiple of 3, making it a monocot. Dicots have multiples of 4 or 5. And the same thing for the petals, unless one of them's fallen off. So 1, 2, 3, 4, 5, 6. So that's 6 petals, multiple of 3. Establishing as a monocot. The same would be true of the other one over there. Okay, so this, by the way, this is in your textbook as well. You can pull it up if you want, but there's a diagram of this in your text. It's also in your notes. I've copied it directly into your notes. But on the outside, there's typically a extra set of petals. This type of flower really doesn't have them. This flower doesn't have them, but they're called sepals. So you can actually see it a lot better on the other one than the other one because it's still covered by the sepals. So the sepals are these petals that are on the outside right here, this covering. When this flower opens, these are going to fold down and out of the way for the petals inside. Sometimes the sepals become petals. So I'm not sure for this exact type of flower. These sepals might become petals. In this flower, that's exactly what's happened. So the sepals, as the flower begins to blossom, they change color and they become petals. So, but it's a protective coloring on the outside, covering on the outside of the flower. So those are the sepals. In terms of the actual male and female part, I'm going to take these petals off. So the purpose of the petals, and this is actually examined in a lot more detail in the video that you're going to watch in block 2. It talks about how these are signaling mechanisms for pollinators. And so they often have regions on them that are highly reflective to ultraviolet light, which we can't see. So we can't see the UV reflecting off of them. But insects, pollinators, typically can see into the UV part of the spectrum. So these are like shiny, flashy in the UV part of the spectrum, which is visible to a lot of pollinating insects, including bees. So bees can see into the ultraviolet part of the spectrum. And often there's a pattern on the petals in ultraviolet that you can't see with the naked eye. In fact, really quick, let me show you some ultraviolet photographs here of flowers. Here's a good example. So on the right-hand side there is what it looks like with the naked eye. And then on the left-hand side there, you can see there's a secondary pattern on the flower that's only visible in UV. So it's only visible with UV reflected light, which would be very bright in UV. It's like a flashy, bright UV being reflected. So that's like a beacon for pollinators to attract them in. It's kind of cool. We can't even see that part of the world. Like it's just like it's invisible to us. Let's see if I can find another cool one. That's pretty cool. This is a pattern on this daisy in ultraviolet. It's cooler when they have them side by side. Yeah, here's another one. There you go. This is like a buttercup or something. Here's an example of a ultraviolet pattern that's on there, like a flashy ultraviolet pattern. It looks black because that's how the camera represents the UV, but it would be like a bright, shiny color on it, just not one that's in the visual spectrum for humans. So anyway, so that's the petals. The petals bring in the pollinators, which are going to help the plants have sex with each other. And these, just leave the male and female parts. Oh, they're just falling right off here. They're on pretty loose. So here we go. We've got an anther and a filament here. They come in groups of 3s on this monocot. If you take one of the anthers, which is the little loose guy on top, and you tap it on a piece of paper, if you've got one of these at home, and you take a look. You know what? All the pollen might have fallen off this already. But usually it leaves a little bit of pollen behind. I think these have been jostled quite a bit, so there's not too much pollen left on these anthers, but they are filled with pollen from the pollen sacs. So that's where the male component is. And when going back to meiosis, we talked about male and female meiosis, this idea that in males you're going to produce 4 sperm, and in females you produce 1 egg and 3 polar bodies. It's not exactly the same in plants. It's similar. For the pollen, it's almost exactly the same. So you can think of the pollen as being like plant sperm. It's very similar. You get 4, usually, I mean, again, it varies from species to species, but you usually get 4 pollen grains per cell that you're starting with when you're doing spermatogenesis, or the equivalent of spermatogenesis. Whereas for the female one, usually you get 1 haploid, and sometimes you get also a diploid cell at the end, and then 1 extra haploid which gets reabsorbed. And the reason for that is that diploid cell actually forms a part of the seed at the end. It's like a secondary thing that provides food for the growing embryo. So it doesn't actually form the embryo itself, but it's the endosperm. It creates a section of the seed that is like the food part of the seed. If you've ever eaten a seed before, which I'm certain you have, of some kind of a seed, the thing that you're primarily eating is the endosperm. You're eating not the embryo part of it. Like, oh, I wish I had a peanut here or something. If you ever break a peanut in half, you can see there's like a little leaf inside of the peanut, a little leaf area. That's the embryo part. But the rest of the peanut is the endosperm, which is just like the food that will become the food for the growing peanut plant. And that's the part that contains all the nutritional value for you and for the plant. It's like basically full of carbohydrates for the plant. So so that's not really a sexual component here. The sexual component is the gamete part, and that's the only part that needs to be sexual because it needs to have the diversity. So that's the growing embryo part. So anyway, we've got the male parts right here, and then the part that's left over in your flower in the center right here is the female part. And obviously, there's a problem with this, which is if you've got your male parts and your female parts next to each other, you're going to get auto-fertilization, right? Self-fertilization, where pollen just goes from the same flower, and that does happen. So some plant species have a problem with that, where they often self-fertilize. They don't always self-fertilize, but they often do. There are various methods in the plant kingdom that they have used to try and avoid this. So some flowers are not perfect flowers like this flower. They don't have male and female parts on the same flower. So they'll only have male parts or only have female parts. So that's one strategy that you can avoid self-fertilization. Some of them have the pollen sacs open at a different time than they have the female part receptive to pollen. So by changing the timing of those two, you prevent self-fertilization. And so, like I said, there's various strategies that they can use to prevent that, but it still occurs. And it's not as bad for plants as it is for animals. So self-fertilization is not bad. In animals, when you get clonal reproduction, it's usually a lot more problematic to have that occur. So anyway, it's not as big of a deal for plants. So looking at the female part here, right at the very top, let's see if I can get a magnification of this so you can see it a bit better. You can use your magnifying lens too if you'd like. This isn't going to work super well. Is that light still on? Is there a light coming out of it? No, it got turned off. Why does this look so bad? Oh, you can actually see there's already some pollen stuck to it on top. That's cool. So this flower has some pollen stuck to that little sticky area on top of it already. So that's the female part of the plant. The very top of it is called the stigma. That's the sticky part. Then right below the stigma is the style. So the style is the tube that goes down. And when pollen lands on the stigma, it actually grows a little section that goes all the way down the style. It's like a little tiny tube that goes all the way down the style into the ovary of the plant. So the ovary of the flower is this bottom part right here. That's the ovary. I'm going to cut into it right now so you can see the eggs inside the ovary, which is kind of cool. You don't really think that there's eggs inside of the plant, but they're in there. I'm going to use a scalpel. You guys can too. Just please be careful with the scalpels again every time you cut flesh. Don't cut towards your own hands. Be extra careful here. Cut myself. Okay, here we go. So cut it in half. There we go. So there's the style coming down into the ovary. Oh, and you can, I don't know, can you guys see the little white circles in there? So those are eggs. They're ovules. Each one of those ovules is going to become a seed. So a pollen tube will grow down from the pollen, down through the style, into one of these ovules, and fertilize it. And like I said, sometimes there's a double fertilization, a second component that will become the endosperm. Not every flowering plant works like that, but I wish this was better focus. I've got to find like the perfect spot. Right there, I think. Anyway, you can see the little white dots. Those are the eggs. The ova. Ovules, rather. So, this ovary, that's the larger structure that all the eggs are sitting in, is what is going to become the fruit. So if you think back to like an apple or a peach or whatever, pick any fruit of your choice, that started off as an ovary for that particular plant. Once it becomes fertilized, these petals are going to drop off, all of the male components drop off, all the style drops off, all that stuff falls off, and then the ovary at the bottom is going to begin to develop into the final fruit. Okay, so the plant is going to pump a bunch of sugar in there, and it's going to grow and enlarge. And the seeds inside, because they become fertilized, are going to start to mature and form a multicellular protoplant, I guess you'd call it. Just a pre-plant, an ungerminated pre-plant. Okay, a seedling, essentially, that has not emerged from the seed yet. Okay? So those are going to begin to grow in there. If you eat an apple or something, you've seen the seeds in the middle. So those were the eggs that were in the center, and then the apple part is the ovary, the outside part, which then becomes your fruit. Okay? That's a true fruit. So there are things that we call fruits that are not actually fruit because they're not made out of the ovary. So true fruits are made out of the ovary. I think there's a question on your quiz about this. But something like a strawberry, for example, is not a true fruit because the seeds there are actually on the outside of it, and the fruit is not made out of the ovary. So every single seed that you see in a strawberry is actually its own true fruit. It is a fruit, and it's on like a complex that's not made out of the ovary of the plant. So that's a little bit confusing. But things that we call berries are often our fruits, like blueberries. A blueberry is a true fruit. The blueberry is made out of the ovary from the flower, or an apple or something like that. Those are all true fruits. Tomatoes are fruits. Anyway, a little bit confusing. I'm going to take a look at the other one just for comparison's sake. I also wanted to mention that if you're looking at something like a dandelion, okay? That's a bit of a different configuration in the flower. So I'm just going to pull one up real quick just so you can see what I'm talking about here. You guys have seen this before. There we go. Oh, beauty. So you can see the male parts sticking up all around the outside right there, right? Those are the long filaments with the anther on top. But if you look in the center area, it's a little bit hard to see. I'll try and zoom in on it here. This part right in the middle are a whole bunch of different female parts, okay? There's a whole bunch of them there. A whole bunch of stigmas that are arranged like in a circle, okay? And every one of those stigmas goes down to its own fruit, or to its own ovary, which will eventually become a fruit, okay? So you guys have seen the end result of this, which is when it matures after it's been pollinated, you get like the fluff at the top, right? And then each piece of fluff has a seed attached at the bottom. That seed is actually a complete ovary with a seed in it at the bottom of every single one. So every time you have a separate female component like this, it has its own ovary at the bottom. So this is an example of a flower that has a whole bunch of female parts in it, not just a single one. So there's different strategies. They don't all look the same. It's when you look at a dandelion, it's not like this flower where it's immediately obvious what's male and female. You have to really look closely in order to see the male and female parts, but they're always there. Okay, so I think that's interesting. Let's take a look at the other one just for reference here. If you guys want to cut into yours here as well, you can do that. I'm gonna lens off here. So this is a again a monocot. These off. I think these are sepals on the outside. Yeah, I think they are. And then underneath there will become the petals. So these are sepals. Oh, actually, maybe not. Is there 6? Then these are all petals then, if there's 6. 2, 3, 4, 5, 6. Yeah, they're all petals. All right. So here we've got. Why does it keep switching the photo? Here we've got the male parts right here. Anther with pollen sacs. They're not mature yet, so if you actually look at the pollen sacs that are on this anther, there's no pollen on it yet, or at least the pollen hasn't emerged. It's not mature yet, so it hasn't, because it has blossomed. The flower obviously doesn't want to drop all the pollen before the blossom opens, so they're not matured yet. And again, if you look at these, there should be 6 of them, because it's a monocot. 1, 2, 3, 4, 5, 6. Good, you didn't let me down. All right, and then here we've got a real classic looking stigma at the top here. Maybe. There we go. Oh, that's a pretty good view. Pads, sticky pads on there for receiving pollen. And then the style, just the tube where the pollen, it's going to grow pollen tubes down. And then at the bottom, we've got an ovary, which eventually is going to become the fruit for the plant. For this one, I'll cut it open. You can see how the eggs are arranged a little bit different in here. There they are. Oh, it's a little bit clearer to see these in here. Let me just get it to the right depth. Oh, shoot, I had it. There we go. Can you guys see them there? They're white and they're lined up right there, like in a row. Shoot, this is never hard to see. I'm doing my best. I gotta get maybe something so I can digitally. Oh, we're so close. They're right there. I mean, you can't see it with the naked eye. There. I don't know if you guys can see this. It's really hard to see. Do you see them? They're like lined up, sort of like little bricks along there. Can you guys see that texture at all? No, it's probably too hard to see.
Like it's a little, like, almost looks like a ladder.
Yeah, like a ladder. They're lined up like a ladder. If you, with your lens here, if you want to zoom in on it, I don't know if you guys can see it at home. Hopefully, I can't tell what's coming through on the video, but I'm hoping you're able to see this. If I put this little platform down here, that'll help. Let's try that here. Wow.
You can kind of club them out.
There we go. There, you can see it right there. That's a little bit clear. Along the top there, it's like a, should look like a little bit like a ladder. So each one of those is a separate egg. Pretty small. So those are going to become the seeds eventually. All right. And that's it, guys. It's not that complicated. These are the sexual organs of a plant. They're not exactly rocket science, but questions about plant flower structure, what the parts are for, what they do? Fairly straightforward. Okay, cool. Did that work okay? Could you guys kind of see what was going on? Okay. Cool. I'm still relatively new at doing that stuff, but we haven't done it this way for, well, it's new. All right. So what you're going to do now is in your note package, page 14, you're going to read the little section on sexual reproduction in flowers. Okay? Then you fill in on the little flower section in your notes package. It just asks you about what the function is of each of the parts that's mentioned. We discussed it together, so you can use that. You can also look it up in the textbook. It's in chapter 13.3. I also placed a link to it under day 17 here, if I refresh it. I just added it, so. Yeah, I added it under day 17. So it's chapter 13.3 in your textbook. It goes over the same information if you're unsure about the function of any of these things. And then what you're going to do is you're going to watch this cool documentary. It's called The Power of Flowers. It's part 2 of a 3-part documentary on the evolution of plants. And it is absolutely fascinating. I highly recommend it. Then you're going to do 602 in your textbook, number 7 and 8. It's just asking about plant structures and functions of the flower specifically in sexual reproduction. And then you can try out the exit quiz. We're okay with that? No one has any questions? Okay, let me know if anything comes up, okay? You're going through stuff, watching the video, you're like, oh, I don't get what this is. What the heck is this thing? Let me know.
We do with all our stuff.
The dissection equipment is, I'm sure, still clean. I'm sure it's fine. You can just put it back in your dissection kit. And then the flower stuff, when you're done with it, you can just put it in the garbage. If you want to leave the paper just up at the front here, the lenses can go back with the lenses. If you want to stack your dissection kits just on this desk, and then the paper you can leave here just in a pile. I'll use it for the next section so it doesn't get wasted. Yes. That part right there.
Actually, what part is that?
Petals are modified. So it is like a new structure. Yeah. You know what? It could be one or two cells that we could throw it under the microscope and look, but I bet you it's so much. So, yeah, pretty thin. No worries at all.